Hey everyone. So I am going to go through this really quick review. I've placed all of the PowerPoint presentations on Canvas so you can go back and review a little bit. I'll just go over the main pieces using my PowerPoint so that you kind of have ideas on what you should be looking at when you're going in and studying. Okay, let's see. Okay, sorry, didn't start where it should. Okay, so the first thing we need to remember is the protein structure or function. Remember that we're looking at the different levels. This is the highest level is the quaternary. Primary is the smaller one. It's the individual amino acids. If we're looking at secondary, we have the pleated sheets and the helix, and then the 3D versions are the tertiaries. There are several different functions of proteins and enzymes. We've talked about this before. So when we're looking at just overall structural pieces to that, there will be, when we're looking at the overall view of how the enzymes work in the body. So remember that if it's over a line, it is an enzyme and it's trying to make your material go from one to the other to be able to create a different type of energy or break it down. So there's protein functions would include enzymes, transport, structural support, cell signaling, and immune deficiency, which we've talked about in the last couple of lectures. Remember that there are biochemical reactions that occur because of enzymes, and those enzymes are also affected by pH and temperature. So you may need a cofactor for some of those to work, or they might just work with themselves. Remember, anything coming in is the reactant, and anything that's leaving that process when it's done is the product. So if we're looking at the chemical pathways, so we're looking at three different types. You're looking at dehydration, which is where it actually pulls water out and joins the material together. You're also looking at hydrolysis, where it's breaking down the polymers. In other words, it's breaking down big chunky things to little things. That is part of the exam that you'll want to go back through. And then if we're looking at metabolic pathways, make sure you go back through the enzyme lecture that has those different pathways that show which enzymes are creating new material from the previous material. And so anything over that arrow is going to be your enzyme, and everything on either side is your product in your material that you're starting with. So make sure that you're looking at that. So remember that when we're looking at an enzyme inhibition, competitive substrates will go in and just click in and take the space up from the substrate that should be there. They do not change the enzyme, but non-competitive or allosteric will actually alter the enzyme shape and make this active site where it will not be available for the substrate to work. When we're looking at membrane transport, there are several different things to remember here. So remember that as far as the actual like transport cell biology, we have ribosomes that are made in the endoplasmic reticulum. The rough ER for initial processing. So remember, the rough ER has ribosomes on it. And then that is sent in a vesicle and given to the Golgi apparatus, which is the Amazon of the cell for modification and sorting. And then it is transported out of the Golgi apparatus, whether it is inside the cell or it is released from the cell using the way of exocytosis. Remember, exocytosis is a cell transport piece that requires energy. So anytime you see a vesicle, you have to have energy in order for that to occur. So when we get to membrane transport, we're looking at different types. There's the passive transport. This is your osmosis and diffusion. So if you're going from a high concentration to a low concentration, that's the direction you're going to use. So diffusion is all things including anything that's going through a channel that does not need energy. So that's your big piece. Facilitated, it uses that protein channel. No ATP is included. It's still considered passive. And then you have active transport, which requires ATP. So that is things that move against the gradient, so we're going from low to high. It also includes things like endocytosis, exocytosis, those vesicles moving things in and out. And with that as well, cell signaling is actually going through the cell membrane. Remember that there is a pathway for those ligands to bind with the receptors. Make sure that you understand or realize that anything that is binded on the outside is hydrophilic, and anything that's used with inside the cell in the extracellular space is hydrophilic binders and receptors. And that triggers that cell response that we talked about, where the ligand hits the receptor sends the signal is translated, the cell does its job, and then it's released. This was this PowerPoint that we went over Ozempic and how that works. So there are different biomolecules that we talked about as well. So we're talking about lipids and fatty acids. So remember that glycoproteins are generally used in cell recognition. Those are the cell surface pieces. There are 3 fatty acids in a triglyceride, and remember that there could be 2 types of lipids on that, either saturated or unsaturated. If we're looking at an unsaturated fatty acid tail, it is going to be something that has a double carbon. And then if we have a saturated, it is going to be that nice little solid carbons all the way across one bond. Nucleotides is the base for DNA. It's the sugar for ribose and deoxyribose. Make sure that you realize that there are 3 parts to that. There's your phosphate group, there's your nitrogenous base, and your RNA base as well. So the DNA structure has that 3 carbon piece, and I'm going to go over this and send you guys out the DNA PowerPoint that your sub is going to cover on Tuesday. I'll send that out today as soon as I'm finished with this one. It has 3 carbons. It has an OH group or a hydroxyl group. And then the 5 end has the phosphate. So if we're looking at the structural piece for DNA, the nucleotide at the top, that 3 carbon or that 5 end carbon has the phosphate, and the lower 3 is the hydroxyl. The base pairs for DNA are adenosine or A to T, C to G. DNA replication happens when the helicase is unwinding and DNA is now available for our DNA polymerase to go in and add nucleotides in order to replicate that. And then remember that the gene expression comes from transcription, not necessarily of actual nucleotides, but it goes from DNA to mRNA. And then DNA is actually wrapped around histones. So for the rest of this, those are the basic pieces for the test. There's also making sure that you are familiar with the vocabulary words. And I am actually posting all of the PowerPoints from each one of those. So I know it's not a very extensive power review, but I am posting the PowerPoints for you to go back and look at. I would definitely take a look at all of the diagrams specifically for cell signaling, which type of cell signaling that it is, either direct, paracrine, or the endocrine. Otherwise, I will see you guys on Monday or Tuesday of next week.